Welcome to neuroanatomy series, and this is episode 7. Last lecture was about action potential. Today we will proceed further. So let's start with the synapse. The nervous system is made up of massive number of neurons that are interconnected with each other. When two neurons come close to each other, these neurons must communicate with each other to function as a system. They form a way to communicate with each other, and this communication is called synapse. Most of neurons have synaptic connections up to 1000 or more other neurons. Under normal physiological conditions, this synaptic communication is unidirectional, means which neuron is sending signal, it will always send signal, and which neuron is taking signal, it will always take signal. This communication will not be bidirectional, means it will be not like that one neuron is sending and receiving signal. However, synapses have many forms, but for now we are just talking about most common form of the synapses, that is axon of one neuron and dendrites of other. This is axon of the neuron. As I said in my previous lectures that axon sends signals. So it is the part of the neuron that sends signals that probably initiated in other neurons and then propagated from the axon to the synapses, and from the synapses are received by this neuron, or and could be the possibility that they initiated in this same neuron. I have described the initiation of the impulses and propagation of the impulses in my previous lecture. You can see if you want to, and link will be in the description. Now, as the axon approaches to synapse, it may have one or more terminal expansions that are called synaptic boutons, and each of the synaptic bouton can make the synaptic contact. A single axon may terminate on a single neuron, or a single axon may synapse with a multiple neurons. In the same way, a dendrites of a single neuron may have synaptic junctions with axons of many different neurons. This is dendrites of a neuron, and these are synaptic spines. These synaptic spines make receptive sites for synaptic contact, and these are called efferent bouts, and those extensions of the axon are called afferent bouts. Synapses are of two types, chemical and electrical, but most of the synapses are of chemical type. They use a chemical substance that is called neurotransmitter. Now this is bouton of the axon, this is bouton of the dendrites, and this is neurotransmitter that is released from the axon. Plasma membrane of this region is called presynaptic membrane, and plasma membrane of this region is called postsynaptic membrane, and this region is called synaptic cleft. These are very important terms to remember. This synaptic cleft is about 20 to 30 nanometer wide. Okay, one important thing to note here that the presynaptic and postsynaptic membrane are thickened from the other plasma membrane of the cell, and the cytoplasm underlying these membranes have increased density. This is because of increased activity of this area in comparison of other area of the cell. The presynaptic portion of the axon contains many small presynaptic vesicles that contain the molecules of neurotransmitters. These vesicles fuse with the presynaptic membrane and discharge the neurotransmitter into the synaptic cleft, and this process is called exocytosis. We have a process that is called endocytosis, where vesicles are taken into the cell. But this process is called exocytosis because the vesicles are taken out of the cell. In the human nervous system, there are many undifferentiated synapses. These can be developed in a differentiated synapses as needed, or the differentiated synapse can undergo atrophy when not needed. This plasticity of synapse have great importance in learning and development of memory. Now back to the topic. The vesicles contain neurotransmitter substance that is released in synaptic cleft. Usually there is one neurotransmitter, but several neurotransmitters may be present. One neurotransmitter acts as principal neurotransmitter, and other neurotransmitters act as modulators that modify the activity of the principal neurotransmitter. For example, acetylcholine is principal neurotransmitter of many neurons in CNS and PNS. Dopamine is principal neurotransmitter of substantia nigra, and glycine is the neurotransmitter of the spinal cord synapses. Now these vesicles have to empty in the synaptic cleft. So suppose a action potential is generated in a neuron, or this neuron has received the action potential from another neuron. That action potential propagated to the end of the axon and it reached this synaptic bout. Now from here this neuron will communicate this action potential to another neuron by the neurotransmitter to tell the another neuron to have or not have the action potential. Now, as the impulse reach here, the calcium channels will open and a rush of calcium ions inside the cell will take place. These vesicles have the sites to attach with the calcium ions. This is a presynaptic vesicle that shows snare complex on one side and synaptotegmin on other side. Synaptotegmin is a protein and snare complex is a group of 24 proteins. Both have the calcium sensing sites and both are said to have a major role in exocytosis of presynaptic vesicle. It is proposed that synaptotegmins promote synaptic vesicle insertion by local bulking of plasma membrane under the synaptic vesicle, which is tethered to plasma membrane by snare complex. After exocytosis, the neurotransmitter is emptied into the synaptic cleft, where it is received by receptor ion channels or receptor proteins of the postsynaptic membrane. These receptor proteins go into the conformational change and open the ion channels in the postsynaptic neuron where the action potential is generated through this conformational change. Note that this neurotransmitter can be inhibitory also, and in this case this is increase the action potential threshold of the postsynaptic neuron. So the normal stimulus which can generate the action potential in normal circumstances will not generate the action potential after action of this neurotransmitter. Now this was the chemical synapse. There is another type of a synapse that is rare in the humans, that is electrical synapse. Electrical synapse is a gate junction that contains the channels that extend from the presynaptic neuron to the postsynaptic neuron. The neurons in the electrical synapse, as name suggests, communicate electrically. Action potential is normally propagated by the ionic current flow as it is propagated in one cell or one neuron. In the chemical synapse, one neuron is a signal giver and another is receiver. So the communication is unidirectional, but in the electrical synapse the communication is bidirectional because this is simple ionic current flow and it can flow in both directions. So both the neurons can give and take the signals, and it is also faster than the chemical synapse. Other less prevalent types of synapse are exosomatic synapse, in which the axon of one neuron is communicating with the body of other neuron. Exonic synapse, in which the axon of one neuron is communicating with the axon of other neuron. And dendritic synapse, in which dendrites of both the neurons are communicating with each other. That's all about today's video. Our neuroanatomy journey is continued, but till the next video, Khuda Hafiz.